All right. Just while everyone's still coming in, I will welcome you all back. I will hope everyone is feeling refreshed and bright and breezy this morning after our lovely conference dinner last night. I didn't get any reports of people lingering in parks at 3 o'clock in the morning this year. Those of you who were in Christchurch last year will know what I'm talking about. So well done, you guys. Just a few announcements today. Our lunch today will be sponsored by Digital Science, so looking forward to that. Our sponsors, who are awesome, who paid for your dinner last night, are all running competitions, and they would like me to remind you to scan your phone on their amazing competitions so you can win things. So you've got the coffee cart's got a scanny thing there, and then all the sponsors have got their QR codes on their booths. So go and scan them. Sorry, Murray, I know you really want to win that Lego. If anyone wins the Lego and doesn't need it, Murray's your man. He'll probably even pay postage. What else we got? There's going to be a survey coming out. We're going to do the post-conference survey. I think you've already got a survey, though, if you were in the research data BOF on Tuesday. There's a survey from that that will be open for quite a while, I think. A wee while. But we'd like you to fill it out if you are so inclined. And I think we will be sending a link to the survey out with the post-conference survey as well. So you'll have plenty of opportunities to do that. So please do that. And that might be it from me. So yes, surveys, please fill them out when you get them. And I'll remind you that this afternoon as well. All right, it is now my great pleasure to hand over to my good friend and collaborator, Professor Chris Brint, who will introduce our keynote this morning. Chris.
Ko ranganui ke runga, ko papa tuanaku ke raro, ko na tangata ke we waina nui, te he mauri ora, tēnā koutou, tēnā tātou katoa. Well, it's a great pleasure today to introduce Kaz Simons, and Kaz is the Rare Disease Program Lead at the Centre for Population Genomics in Australia. The Centre for Population Genomics is a joint initiative between the Garvan Institute and the Murdoch MCRI Children's Research Institute. So Kaz's role there is to really lead national collaborations across Australia, which translate complex genomics into clinical action. And I guess Kaz's work, like what many of us aspire to do, sits at that really exciting interface between the computation, the biology, and the patients. But the cool thing I think about Kaz is Tauranga based. So Kaz is living this dream that many of us would love to live but haven't quite achieved, of having international impact from New Zealand. So Kaz has a PhD in computational biology and genomics. Kaz has spent a long time in Queensland running research groups at the IMB and the University of Queensland, and also at the Murdoch in Melbourne. Kaz was previously also head of computational biology at the Queensland Facility for Applied Bioinformatics, which many of us have had interactions with. I think Kaz's talk today fits incredibly well with the conference theme this year, Are You Ready? Since I think Kaz's talk touches all four aspects of that theme, all four pillars: the community, the partnership, the exciting cutting edge and innovation. So the title of Kaz's talk is Making the Obvious Easy, How Collaboration and AI Are Removing Bottlenecks in Rare Disease Diagnosis and Discovery. Please join me in welcoming Kaz.
Thanks, Chris. Thank you, Chris, and thank you for the invitation to speak today. First of all, I want to apologise for my lofty title. Uses a colon, always a bad idea. It was a bit of a last-minute invite, and Kristen asked me, any chance we can get a title by tomorrow? At this point, it was very late at night in a hotel room, and I was travelling, and somehow I thought this was lofty title was going to be a good idea. Anyway, I want to start my talk by introducing you to Abby. So Abby, who's now 14 years old, was born, and at around 3 months of age, her mother started noticing that she was missing some of her developmental milestones. When she was around 1.5 years old, she started having seizures. These were quite severe and have been largely uncontrolled for her whole life. She's been put on a large number of medications trying to control her epilepsy, each of which has failed. She's now a bright, happy 14-year-old girl who's non-verbal and has a profound intellectual disability. Her family have been really driven to understand Abby's condition and try and find a genetic diagnosis that's causing that disorder. They, working with her doctors, put her through successive rounds of genetic testing that really followed the arc of the development of genomic medicine, starting with array-based tests when she was young, moving into genomic sequencing through successive rounds, none of which found a result. Her family has then enrolled her in every research program that they could access, in the hope that some research study would help find a diagnosis for her. Essentially, her and her family have been waiting her whole life for a diagnosis. On the right, I'm just showing a couple of quotes from her mother, Abby's mother, that she gave me permission to use. The first, that she'd never given up hope. She didn't think they were going to, thought they would get a diagnosis eventually, but at this point, she didn't think it was going to be anytime soon. And the really important one, I think, is the second one here, which is a message we hear from so many of our families affected by rare conditions, that she felt being a lone voice in the dark, it's bloody scary. So my research focuses on the diagnosis of rare genetic disorders. What is a rare genetic disorder? It has this fluffy definition that changes by district. In Australia, it's defined as affecting less than 5 individuals in 10,000 people. There are a whole range of estimates about how many people are affected by or are living with rare disease. Some of the more recent ones are suggesting it's around 2 million Australians. Apologise, all of my numbers here are Australian-centric. Obviously, this applies all around the world. Over 80% of rare disorders are believed to be genetic in origin, and the average time it's taking for somebody in Australia to get a diagnosis at the moment is around 4.8 years. So from that's the time where the clinicians recognise this as a disorder or a disease, and they want to give them be able to give them a diagnosis. It's often taking years. One of the challenges of rare disease is that there's not one thing, right? So we talk about these disorders, they are individually rare, they affect very small numbers of people, but they are collectively common. And the other aspect here is there is a massive diversity in these disorders. So over 6,000 genes have been implicated in monogenic disorders, so this means that you can have 1 or 2 genetic variants in a single gene, and that can result in a rare disease. The last 15, 20 years, we've seen this explosion in genomic technologies, our ability to sequence genomes and investigate the genetics of individuals. And this has been really amazing for both research and diagnosis in rare disease. However, the tragedy is that still, even with all those advancements, something like half of all individuals tested still go undiagnosed at this point in time. And that's a number I don't think we should be living with. So people sometimes talk about having a diagnosis as the end point of what we'd call a diagnostic odyssey, right? So, hey, yay, great, you've got a disease. But it's really important to know that that's not true. The diagnosis is often the beginning of the journey. For families living with rare conditions, it's often the start. It changes what care they can be provided by their clinicians. It changes what a family can plan for, and it stops a huge amount of costly waste and often invasive investigations of that affected individual. At the population level, each diagnosis also strengthens the evidence base that makes the next diagnosis easier. So it's a cumulative thing. The more people we can diagnose, it makes the next diagnosis easier to make and therefore faster, and therefore we can get answers to these families sooner. So what are the elements of a genetic diagnosis? Given our diverse audience, I'm not actually going to step through the nuts and bolts of this here. I'm going to keep this fairly high level. But essentially, what we can do now is we can take a sample of blood or tissue from an individual, we can extract the DNA from that, and we can sequence that and use that genetic sequence to identify all of the individual genetic differences in that person's genome that makes them unique. So these are all of the variants that we all have within us that make us a person, and we can do that for the individual and perhaps their parents as well, or other members of their family. From there, it's basically a needle in a haystack problem, right? So we can generate that list of all variants that make that individual unique, and this is typically a number of the millions, and that's now relatively routine and something that technology has given us and we can do well. The hard part then is narrowing down that list from millions of variants down to the one or sometimes two changes that actually explain that disease. To do that, we need to be able to demonstrate that variant or candidate variant is plausibly damaging to the gene, so it's actually going to impact the function of the gene that it's within. We need to know that the gene is genuinely linked to the disease. We need to have seen that it's present in other individuals with that same disorder. We need to understand where that variant has been seen before. So we need to know if it's common in healthy individuals. If it's a variant is common in healthy individuals, then we can rule it out because it's very unlikely to be the cause of a rare disease. However, if that variant's been seen only in people with a similar disorder, then that gives us very strong evidence that this might be the cause of their disease. And that's really the process that we go through. And what that is also, we look at those individual bits of evidence, and it's not a binary decision, but it's really a burden of proof situation. So we've got to assess the different sets of evidence we have and see whether we can meet that burden of proof that this variant or variants is very likely to cause that genetic disorder, in which case we can then return that as a diagnosis to the individual. If we can't meet that burden of proof, then we can't return the diagnosis. So why do we have this approximately 50% of individuals that we can't diagnose? What's the basis of that? Well, it falls into essentially two large buckets. It's not a single failure mode. So some of those cases are genuinely beyond current knowledge. They need new discovery to be made to be able to turn that from something we don't know about to something we can and provide a diagnosis. Others are diagnosable in hindsight, but the methods, capacity, and the way or what we're doing with their data or when different pieces of information become available mean that we fail to operationalize that and bring those pieces together to cause a diagnosis. I'm going to show you some of our work that's targeting both aspects of these problems. But early in my career, I was really focused up here in this discovery gap and really interested in finding those novel disease genes. The further on I go in my career, I think more and more about this delivery gap, and it's what keeps me up at night, both from my own research cohorts, thinking about, hey, is there information out there that actually I could be giving one of my participants a diagnosis, but I just haven't seen that? But also as a system as a whole, this is something we need to be doing a better job at. So, shifting gears a little bit, I'm going to show you that rare disease diagnosis loves research. This is not a surprise. So if we think about a typical patient journey at the moment, they'll start out and working with their doctors, they'll recognise perhaps that this patient might have a genetic disorder. Hopefully they'll have access to genomic testing, which means that sample in a clinical setting, which means that a sample of DNA can be taken and it can be sent off for analysis. And the happy path is that family receives a diagnosis and that can go on to influence their care. from that point. However, like we've discussed, half the time that's not true. These individuals won't initially get a diagnosis from the genomic testing. So one of the best things we can do for those individuals is offer them the opportunity to participate in research analysis looking into their disorder. So this means there are dozens, hundreds, thousands of research groups around the world who are interested in the challenges of different rare disorders, where they can take that data or the genomic data that was either generated from that clinical test or applying new fancy genomic analysis methods, like you might have seen Leah's talk yesterday, talking about long-read sequencing, that can move us forward. And they can apply those new techniques along with updated knowledge as it evolves, and hopefully convert that into diagnoses. This is obviously a really great path and something I've been involved with for a long time now. However, again, one of those things I think about now, am I the solution or am I sometimes just the bottleneck? Just to give you a small anecdote of this, I'm going to take us back to 2012. To fix that point in time, I've given you Psy here, hopefully you can remember, dancing along to Gangnam Style. And I'm going to introduce you to Adeline van der Veer. Adeline is a paediatric neurologist, currently based at the Children's Hospital of Philadelphia. Amazing, fantastic person and a rare disease researcher. Her specialty is looking at white matter disorders of the brain, sometimes known as leukodystrophies. And so these are a set of disorders that affect how the white matter in the brain develops and grows over time. One of the things that she was a world expert in, it was in not just working with her patients, but also working really carefully with imaging data from MRIs. She would take MRI imaging from each of her patients, and they would look through that with a fine-tooth comb, not only establishing, yes, the white matter is developed abnormally, but looking for patterns of exactly which part of the brain are affected, in which order, in which developmental time points, and use that to group together sets of patients that have similar patterns in their imaging. So she had knowledge of genetics, she had access to a great cohort of patients, she had the expertise to put these groups together that were just sitting there ready to go for genomic analysis. But that's kind of where she got to, and she reached a bit of a sticking point because she wasn't an expert in bioinformatics or high-throughput genomics. She'd had a few goes at trying to develop skills within her own group, that's a challenge, or with working with others with limited success. And through serendipity, managed to form a relationship with some young upstart postdoc on the other side of the world, and decided that they could work together. This was a great thing for me as a sort of a relatively recent entrant into the rare disease space, had some of the skills in bioinformatics and genomics, didn't have access to any good patient cohorts. Adeline sent me to start with a cohort of 8 families. She said, sequence these ones first. I'm sure they all have the exact same disorder. I sequenced them. And so I was looking for variants in a single gene, and I was expecting to, have to travel for this haystack. It turned out to be one of the easiest analyses I'd ever done. I looked at all 8, opened up my sort of reports I was generating at the time, and I found all 8 not only had de novo mutations in the same gene, so a new genetic variant that was unique in that affected individual, but they all 8 had the exact same base or nucleotide changed. So a single letter C to T in their genome, and all 8 was the same. Shooting fish in a barrel, I was convinced that rare disease research was easy, and I was going to dominate. This led to a really lovely publication describing the gene TUB4A, which is now known as one of the more common causes for leukodystrophies, and has been, had a lot of impact over time, being able to provide diagnoses for thousands of individuals. More importantly, this was the start of a really beautiful collaboration. So 14 years, 35 papers, and something like 8 novel disease genes later, Adeline and I are still collaborating. It's obvious she has aged more gracefully than I have. But it's been a really wonderful collaboration that's really the best of bringing two different expertises together. But I do often think, should a future Adeline actually need to wait for a future Cass, right? Like, should she be forced to develop skills in high throughput genomics to be able to drive her research forward? She had all of the questions, she knew where to look, she was just limited by her ability to work with the data. And so that brings me to the first thing I'm going to say is an obvious thing we need to be able to move forward, and that is we need to be able to enable and support collaborative research of rare disease amongst this really wide group of rare disease researchers, just like Adeline. And they need to be able to do that without needing to invest heavily in the ability to do the genomics themselves. So to address that, the CPG, we've developed what we call the Cardinal platform. And it starts with a rare disease study. And so this might be just like Adeline's study, or one of a number across Australia, where say a clinician researcher has developed a cohort of undiagnosed individuals. They've recruited them into a research study and they've begun data collection. So that might be by accessing the already available clinical genomic data from the negative test, or it might be through generating their own. We then have a process where we work with them to take that data and we make it as easy as possible from them, either working from where that data is archived or working directly with the sequencing facilities and clinical labs to have that data transferred to us. And then we take de-identified metadata about those individuals, basic information about family structures, and information about the symptoms or the phenotype of the individuals, and we bring that into our Cardinal platform. The data is then analysed through a set of standardised analysis pipelines that we run, looking at all of the common types of variants that a researcher may be interested in assessing, and it's also capable of bringing in traditional short-read data, which is the backbone of most genomic testing at the moment, as well as the more recent long-read data and increasingly RNA-seq data as well to complement the genomic data. That data is processed and harmonised and entered into our Cardinal data store, and from there we make it available back to the researchers for a collaborative investigation of that data via the Cardinal analysis portal. This portal is built on the backbone of Seeker, which is a wonderful product, open source product released by the Broad Institute, who we also collaborate with. And it is a really accessible, user-accessible interface that is secure with good data permission capabilities that allow these research groups to access the genomic data, use really flexible but intuitive filtering strategies to do that needle in a haystack approach, and be able to, hopefully, generate a diagnosis for their individuals. I also have the benefit of having a team of 4 variant analysts who, in my team, who work with each of these research groups and support them, help them use the tools, and really allow them to get the best out of the system. Some don't need our help at all because they're running fast. Some need the help in understanding how this type of work, and these analysts are there to help them do that. This results in diagnoses for the individuals, but also this really important gene discovery aspect, which feeds into more diagnoses around the world, and also gives us a platform where we can embark on safe data sharing with both within the cohort, but then across with other sites around the world as well. One of the advantages of having this lovely harmonised data store where all of these patients are processed in the same way is it allows us to also do things across this whole meta cohort. So we can provide tools like I'm going to talk to later in my presentation about automated reanalysis, where we can go back to each of those individuals every month and relook at them again in an automated fashion, trying to see if there's some updated knowledge or new discovery that would be applicable to them, and present that to the collaborators for evaluation. Equally, we can do cohort-wide analyses, for example, say, hey, look, we recognise there's this new thing in 6 different patients across, say, 4 different projects. You guys should get together and talk and say, is this perhaps a novel disease gene or a novel finding? All of that information going back to the collaborators for further investigation. What does it look like? This is really hard to do in a screenshot, so we won't dwell on this, but essentially this is and it'll give you an idea of a really nice intuitive user interface that provides this needle in a haystack function, filtering function, ability to get right down to the variant level, see the information that these analysts will need to actually determine is that variant and gene potentially causative of disease. It adds a set of features for being able to tag variants and add notes so that those teams can work together and track the analyses over time, and also does things like integrates ability to go right back to the raw read level, so you can review those variants and ensure that you're actually looking at a real variant and not, say, a artifact of sequencing or variant calling process. Cardinal's now grown into a really fabulous collaborative network. We're now supporting over 40 research cohorts from mostly Australia, but also a few outside of Australia. And it consists of over 10,000 individuals from over 6,000 different families. The system supports or has data from over 8,000 whole genome sequences, several thousand exome sequences, and a building number of hundreds of long-read sequence data as well, and covers a wide gamut of rare disease research areas, and these are just some of the PIs or cohort leads from across Australia, and this is really a who's who of rare disease research in Australia that we're very lucky to work with. So, just to give an example of the power of a system like this, I'm going to use the anecdote of a Slack message we got one morning, just under 2 years ago, from a collaborator in the UK called Nikki Whiffen, who's a really fabulous rare disease researcher at Oxford. She'd been working in the Genomics England data set, which is a very large data set of undiagnosed individuals from the UK. And she came to us and said, this new gene, RNU42, we've seen it's likely to cause a neurological disorder in Genomics England data. Can you replicate this in Cardinal? Within 30 minutes, I was able to go from that Slack message to being able to look across all of our cohorts in Cardinal, identify 6 patients with RNU42 mutations from 4 different projects. That morning, I was able to connect all of the cohort owners from those 4 different projects with Nikki and ask them if they'd like to collaborate in the research study describing this new gene. And this resulted in a Nature paper published recently last year. Novel gene, Nature paper, this doesn't quite make sense. The missing thing here is that RNU42 is a non-coding RNA, and not only is it a non-coding RNA, it is one of the, we now know in hindsight, it is one of the most common causes of intellectual disability, probably accounting for several percent of individuals with intellectual disability. Why hadn't it been found earlier? Mostly because of the biases built into our systems that avoid looking in non-coding parts of the genome. Unforgivable in some ways, great, we finally found it. Coming back to Abby, who I introduced you to at the start of the seminar, Abby's condition is caused by this variant in RNA42, and she was one of these 6 individuals. So this finding gave her a diagnosis. The families affected by this disease got together and decided to give it the name of Renew syndrome, and now it's recognised as one of the most common causes of developmental delay, estimated to affect over 100,000 people globally. This discovery has allowed a worldwide community of patients and families to come together and support each other, and also triggered a massive amount of research. There are already mouse and MAVE models have been generated for RNA42, and we know of at least 2 pharmaceutical companies that are working actively on therapeutic development. Also, it's highlighted this important new class of pathogenic mutations in non-coding RNAs, triggering a cascade of discoveries in other RNU genes. I think we're up to 3 or 4 other gene discoveries since this time. This is great. When we make a discovery like this, though, one of the first things you think about is, hey, great, I should now go back and look at all of my other cases and see which of them have this RNU variants in this RNU42. We want to do this for the and for something, a big splash finding like this. Maybe that's going to happen because everyone's going to be excited and sort of manually go and do that. But the reality is novel disease genes are being published on a weekly basis. I think there were close to 150 new genes described last year. And many of those don't make this big splash and don't have people sort of going back and doing the work manually to look for them. So, obvious thing number 2. When a new discovery is made, we should make it really easy to go back and review past cases and see where that's important for providing a diagnosis for them. And this is really demonstrated by this anecdote of a single case in one of our studies, who presented with a neurodevelopmental delay early in childhood, and they were luckily enough to be able to access whole genome clinical sequencing in April of 2022, and unfortunately no diagnosis was found from that clinical testing. 1 month later, in May of 2022, there was a publication demonstrating that this novel disease gene, CTR9, is associated with neurodevelopmental delay. What we didn't know at the time is our patient here, shown in blue, their disease was caused by variants in CTR9. Because of the way clinical testing works, it's a point-in-time test. There's no way to take that finding and go back and apply it to previous cases normally. And so, unfortunately, time just marched on. That patient was left without a diagnosis. This is something we can do. We can do something about this. So, what's the better model? Instead of the current situation where we just have, at least in the clinical setting, this 50% of undiagnosed cases essentially get put on the shelf, maybe never to look at again. We would suggest a model where these patients should enter some sort of continuous reanalysis, where we can apply these updated knowledge and maybe even improved methods to reanalyze that existing genomic data. We've already done the expensive bit of generating that data and get them towards a diagnosis. This is not a novel idea, not invented by me or my collaborators, that's for sure. And there's a long history of over 40 published studies now covering over 10,000 cohorts of 10,000 patients, demonstrating that this is not only a good thing to do, but it's probably the single best thing you can do for an undiagnosed patient, giving a diagnosis yield of around 15%. That number is heavily influenced by when the initial testing was done, what type of testing was the time between then and now, and the technologies used, but the message is clear. Reanalysis is really powerful. The problem with each of these published studies is that they generally are single large efforts where a team gets together and say, right, well, I'm going to undergo a reanalysis, and they manually, to a large extent, go through each of those cases and reanalyze them using a method similar to they would for the initial genomic testing. This is very high quality, produces good results, but it's not scalable, and it's obviously hugely expensive given the expertise required to undergo this review. I don't know what that is. So Zinitza Stark is a clinical geneticist based at the Victorian Clinical Genetics Service in Australia, probably Australia's largest genomic sequencing test provider. And she had this vision of we need to develop, apply, and then evaluate an automated platform to repeatedly reanalyze genomic data at scale. And so put together a consortium of individuals, applied for a grant that we were luckily funded by the MRFF. And then it was handed to my team to actually build this tool. So I'd like to introduce you to Telos, the tool we developed for automated iterative reanalysis of genomic data that's really intended to be applied in this clinical setting. So it returns only clinically relevant variants, so it's not a hypothesis generation tool, it's not in that research space, it's really intended for this clinical application. And it's really focused on the scalability factor in the sense that the worst thing we could do is overwhelm the clinical teams with too many variants to review, otherwise they just don't have the time to do that. So the intent is to give them a very focused list, very high specificity of candidate variants that are likely to cause a genetic diagnosis. And we need to be able to run this on a regular schedule. And so we can do this, we can suck up all of the new discoveries that have been made over time and in a timely manner, apply those across all of our cohorts, and then return those candidates to the teams for evaluation. In Greek mythology, Telos was a giant automaton or robot who protected the island of Crete by circling the island 3 times a day, so we thought this was a fairly fitting name. And I particularly want to call out the efforts of Matt Welland, shown here at the top, who's the lead developer of Telos, earning him the nickname of Hephaestus, who was the Greek inventor who invented the automaton, or built the automaton of Telos. How does Telos work? I'm going to skip through this very quickly, but essentially the inputs are information that should be available at the end of genomic testing. So what we call a VCF file, which is essentially a list of the genomic variants held by that individual or plus or minus additional family members. We need some pedigree information, so that just basically tells us who's affected and what their relationship is. And we need some basic information about the presentation or symptoms of that individual, and we encode those in an ontology called HPO terms. That data is processed by an initial variant annotation workflow that essentially goes through a data normalization and reshaping process and performs a set of annotations that are the things that are not likely to change much over time. So the things that we can do once and be happy with and move on. We run this using Google Cloud Compute. Give you an idea of cost. It cost us around 2 cents per sample to run to get to this point. So very, very cheap. We then move into the monthly iterative cycle where we run this variant prioritization workflow. This is where we introduce the dynamic annotation, or all of the data sources that are changing over time, and we retrieve them at the point in time that we run the analysis, so we're working with the very latest data. This applies the gene-disease relationships, any variant disease associations that are known, and applies a set of variant logic modules that essentially automate that needle in the haystack filtering down to give this very specific set of candidate variants that we believe are likely to be the cause of the disease. This step, again, in GCP speak, costs us less than 0.1 cent per sample per month, or around 1 cent per sample per year to be able to run this on a monthly basis. So can run on any system, but this shows that it's really resource is not limited in any way here. This generates the monthly reports that we provide to our teams that then can go through them. And so it's not intended that this is the answer that we just send straight back to the patients, but the intent is for this to go back to the clinical lab who's done the testing. They can undergo that manual review and, where appropriate, do a full reassessment of that individual and the variant and clinical report that clinically where appropriate. Again, I won't, this is just a screenshot of sort of one of the reports and the way it can be used. So either sort of a basic explorable HTML report, or those results can be directly or given in a machine readable format that can be introduced into whatever the variance analysis system that your team likes to use. So for example, we can inject that straight into Seeker so that information is available there within the cardinal context. So does it work? Anecdotally, coming back to our case from the start of the TELOS, this individual had gone undiagnosed for multiple years. They entered the reanalysis project here in October of 2024. That month we ran TELOS, identified the candidate variant in the novel gene, and that clinical report was issued and given back to the family. Works great on a single individual. To look at that slightly more broadly, we evaluated TELOS in two what we'd call gold standard evaluation cohorts that have been studied very extensively. One from the acute care genomics study led by VCGS in Australia, and another from the Rare Genomes Project at the Broad, which is quite a well-respected rare genome study. What we see is that when we run TELOS across these very well-studied cohorts, it returns around 1.3 variants for every trio. So for every family that we analysed, it was only identifying just over one candidate for review. So very small number, very specific, which was exactly what we're after. Within that, TELOS was detecting 89% of all of what we'd call in-scope diagnoses, so the types of variants that TELOS was intended to detect. So really good numbers. We weren't hitting 100%, but that was by design because we were really, this tool is intended to make sure there are no low-hanging fruit left, and while maintaining this very high specificity to not overwhelm curators with variants to review. When we reanalyzed that same data, but just using singletons, so we would take the unaffected parents out of the mix and analyze the affected individuals only, the detection rate fell to 80%, and the number of variants returned increased slightly to 1.8, which was very acceptable numbers as far as we're concerned. Where it really gets interesting is when we moved that into a prospective analysis study. So we ended up studying, prospectively analysing a cohort of just under 5,000 individuals that were from a mix of Australian genomics flagship studies that had been generated over the past 10 years, and then the set of VCGS clinical genomic tests that were returned as undiagnosed from 2019 to 2022. Doing that, and so this at the coalface of this was Zanitza and a very skilled variant analyst, variant curator called Paul, shown here, and they analysed each of the candidates generated by TELOS, and we were allowed to identify just under 250 new diagnoses, giving us an additional diagnostic yield of about 5.2%, which we were very happy about. I won't go into the details, but we're able to break those new diagnoses down by the reason why the diagnosis hadn't been made previously. So some of these were because of there was new gene-disease association, for example. Some were variant reclassification, so essentially where the rules governing that burden of proof had changed over time, and there was a movement for a variant that previously would have been considered not to have met that evidence level, now has gone above it. And there was also an uncomfortably large number of variants that where that diagnosis probably actually should have been able to be made in the original test, but it was missed for various systematic reasons. They were typically in some of the older tests, sort of going back to the 2019 area, where process improvements over time have already caught those holes in the clinical analysis pipelines, and so hopefully wouldn't be happening now in modern day testing. Doesn't end there. We're continuing to run this and expand that cohort over time. So we now have over 11,000 unsolved cases that are undergoing this monthly reanalysis using Telos within the VCGS and Cardinal systems, and that's yielded well over 300 diagnoses before. The other great thing, we're making this available open source, and we're starting to see uptake in other labs and research centres around the world, so this is this impact is not just localised to what we're doing in Australia. The really cool thing is this is not just the point-in-time reanalysis that generated most of those results I showed you on the first page, but this ability to go to the scalable iterative reanalysis. So again, just an anecdotal example. We have an individual here who, in this case, they were an adult presenting with cardiomyopathy who underwent whole genome testing in August of 2020 without any diagnosis. They entered the reanalysis program in July of 2023, and we started running Telos. It didn't give any results. We kept running it for several months in a row, no results found. In December of that year, ClinGen, who's one of the large peak bodies in this area, upgraded a gene called APLK3 for its association with cardiomyopathy in a dominant form. Telos received the information for this upgraded status of this gene, applied that in February 2024. I think our team must have gone on holiday in January, so that giving the information for that diagnosis that could be returned for the individual. The really cool thing about this is how scalable it is. Once we get to this iterative phase, we're only returning around 5 variants for review for every 1,000 cases. So that means like once a month, if you've got 1,000 cases in your cohort, you just get an email notification with just 5 variants to look at, and maybe one of those might be a causative gene. Really, really scalable and feasible to integrate into everyday practice. So of our diagnoses, so far about 6% have come from this iterative monthly cycle. And what's really neat is this number here, which is 32 days is the mean number of time between the new knowledge being disclosed, for example, that paper being published in a journal, filtering through the system where that's been available to Telos and getting to a diagnosis. That is a remarkable number that I'm really quite proud of. And my clicker has stopped working. Okay, so now coming back to our story of RNU42, I can show you how Cardinal and Telos work together in a really nice synergy. So we had this story where we end up with this Nature publication demonstrating this new gene. That triggers RNU42 to be put into the catalogue of known disease genes that Telos works with. So within 1 month of this publication hitting, using that Telos pipeline, it was able to identify 4 additional cases that we didn't know about in the clinical cohort that was unavailable for the research analyses, and diagnostic reports could be issued to each of those families. Give an example of that. Here's a patient who had genomic testing early in 2020 with no diagnoses. This paper comes out in mid-2024, and we can get them that diagnosis within a month's time. So this is really showing Telos doing what it should. The bit I want to think about now, though, is, hey, this is great. We have these new disease genes. We should be able to apply that. Telos should be able to look at each of those new disease genes as soon as it's published and apply that historically. That should be easy, right? Because we have a community, have a good, robust database of all of the disease genes that have been associated with disease, don't we? We keep that up to date? No. Science has a whole lot of failure modes, and this is one of them, where the researchers doing the work, who publish the papers, providing the evidence of association between a genetic disorder and a disease, that's published in the literature. There is no direct connection between those publications and them being coalesced into a catalogue of disease genes that is then used by clinical testing platforms. So each of the diagnostic clinical labs have a reference list of genes associated with the disease, and they only look at those disease genes. And so if you're not in the research setting, in the clinical setting, you're limited to looking at only that set of genes that has met that burden of proof, of evidence that it's associated with disease. There are several initiatives around the world with a mandate of trying to attack this problem and trying to build these catalogues. Some, like ClinGen, are doing an amazing job of doing going through an extremely robust review process of evaluating the literature for each gene and assessing them and providing very strong evidence for gene disease associations and all of the details of that. But the challenges with that model is that it's necessarily slow and manual and can't keep up with the literature as it's published. There are other initiatives, and the one I'm going to talk about is Panel App Australia, again led by our friends in ITSA. And so this is essentially the gene catalogue that's used by many of the clinical labs in Australia, and they have a dedicated collaborative team of mostly clinical geneticists who they really see as their mandate trying to keep this as up to date as possible. They've done this for several years now, and the way they do that has been to monitor a set of usual suspect journals where this type of information is published, and they look at all of the titles and abstracts, identify which papers are relevant, review them on a monthly, and do this very religiously on a monthly basis, trying to enter in this information into the Panel App system, as well as bringing in any information they can find from other sources. So, for example, the other gene curation systems out around the world. This is a critical part of Telos because this is the information source for Telos users. We've chosen to use Panel App simply because it is the one out of all of the ones that really tries to keep a pace of the literature as fast as possible, meeting that mandate of trying to bring the time to diagnosis down. This is just a little bit about what Panel App looks like, and essentially it's divided into gene panels which are associated with different types of disease, and each gene is reviewed by this expert curator, and the evidence is put into this database, and then based on this evidence and a rubric for assessing that evidence, each gene is given a score of green, where there's a high level of evidence for that gene disease association, amber for a moderate level, or red for a low level of evidence. In a clinical setting, only these green genes are used. So to get a clinical diagnosis, that evidence need to be high enough to be given a green. Amber and red, you can best think of as really good research candidates where there's some evidence and we really need to add a little bit more evidence to bring them across the line to get up to green. This sounds great, but the reality is there's around 120,000 articles published in PubMed each month. Obviously, not all of these are related to human genomics, but it gives you a sense of the scale. Up until now, the manual process by this team of half a dozen expert curators had been trying to do their best to keep apprised of this 120,000 articles a month, pull from that the ones that are relevant, and enter that data into Panel App. They've been doing a great job at that, but it's obviously a system that's going to have flaws. So, under the banner of a collaborative project that CPG leads, which is the Australian Alliance for Secure Genomics and AI and Rare Disease, nice backronym of ASGARD, we're interested in building a system to actually do this in a more comprehensive way. So the idea being that we have these 120,000 articles each month. That's pushed into an LLM-based system that can review each of those, decides which ones are relevant, and then go that extra step to look at those papers in detail, extract the information that's relevant for the review, and present that to the curators in a way to make it really easy for them to do the job of deciding what is new evidence and it's not, but also importantly being able to then verify that in the original publications and make that a final assessment of is this relevant, should it be included in a panel app, and what is the level of evidence being provided. So this work has really been driven by a really amazing partnership between Zanitza and a fantastic engineer, Leo, who are both based at VCGS, and Simon, so Leo's working in Simon's team at VCGS. And so this is their work I'm presenting, but it's related to this larger banner of work that we're doing in this space at the moment. So what does this look like? Again, screenshots don't do great justice, but Leo's running this system every 2 weeks now, because to keep up with that large number of papers. And what it generates is a report of genes that it says, hey, for example, this one is showing, here's the set of new genes I think this month probably should be considered as new green genes. An analyst or the curator can go and look at those, they can click in, and they can see the extra detail describing the summary of evidence from all of the literature that's been found supporting that. Sorry, one thing I forgot to mention, what the system does is it looks in this last 2 weeks of literature, identifies any new information. If it finds a paper relevant to a new gene, it then does a historical search going backwards, identifying all of the literature relevant for that gene, and if it's a size number of tractable number of papers, we'll reassess all of that literature and summarize the evidence from all of that literature into this one report. The analysts can then dig in further, looking at each of these parts of evidence and trying to understand at the individual paper level the number of affected individuals, what their variants were, what the modes of inheritance were, things like that. And then importantly, click through from individual points of evidence in this report back to the PDF with a highlighted section of the PDF showing where that claim was actually being made from. This system, so this is a quick graph just showing the over time, so this is mostly from last year, each month, the number of new genes being added to panel app, and the red, orange, and green here showing relating to the status of those of the evidence that they were awarded. So this is pre-AI using the historic system of manual curation. We can see here the bump once the AI-based or LLM-based system comes into effect, showing the number of genes increasing dramatically. So obviously this is slightly stochastic in terms of its numbers of genes per month and may trail off, but you can see that this is giving the curators a lot more relevant, discovering a lot more of the relevant genes and information. What's really interesting though is not so much these new genes, which were never listed in panel app before, but is these what we call upgrades. So these genes were previously amber, this limited set of evidence, but not enough to make a clinical diagnosis from. And the system is pulling out that next little bit of information needed to get these genes across the line, and so we can give a diagnosis from. So this bit has been in effect or working since I think about starting in October last year, and we're already seeing this coming straight through into Telos and giving new clinical diagnoses based on this now. So we're really excited about this, and this is just part of a series of work that we have underway in the space, really looking at making better use of our literature to be able to support the analysis in both the clinical and research setting for rare disease. Some of the things that we're working on is making tailored evidence summaries for different between the clinical and research settings. So the research where we really are interested in that hypothesis generation side of things as well. We have a parallel effort looking at variant disease evidence aggregation, so looking at individual variants and when they've been reported in the literature in the past. And of course, it's very high on our list to get this type of evidence summaries integrated into both Telos and Cardinal directly. So finally, I hope I've shown you a few of the things that we're working on that's really helping me sleep at night. And if you're working in the space or know somebody who is, they may be able to help them sleep at night as well. So Cardinal is accessible and available for collaborations. We're also interested in talking with Kristen about how we can support a similar effort here in Australia. Telos is an open source system available to download from GitHub, can run in any environment, and we really encourage people to work on that. The ASGAD related tools aren't available at the moment, but they will be shortly. At the moment, I'm pointing you to Panel App where you can make use of that information. Finally, I just want to thank a huge number of people. CPG is a team of about 50 people that work together in a really collective sense. I can't name all of them, but their support in making the systems happen is absolute. In terms of the Telos work, this was a wonderful collaboration between CPG, VCGS, the Broad, and Microsoft Research. And so there's a large number of people to thank there. And finally, the CPG's rare disease team has contributed to everything you've seen here. And then a particular thanks to Zinitza, Simon, Leo, and Paul from the VCGS team across both the Telos and ASGAD data. And with that, thank you very much for listening.
Thank you very much, Kaz. I think that is an incredible example of reducing to practice a lot of what we think about. We've got time for a couple of questions. Maybe if I can kick off.
Sure.
Does this change how we train clinical genetic scientists and clinicians?
I believe it should.
Yeah.
So mic'd up. Yes, so I don't believe definitely a clinical geneticist shouldn't need to be doing this day in, day out, but they should absolutely have the experience of going through this process and understanding what is possible. And the clinical geneticists I work with now, some are very engaged and want to get right down to that deep level and be able to participate. Others are very happy to be hands-off and let the curators do their jobs. But at least having that experience and being able to get their hands dirty doing the analysis, I think, is an essential experience. Yeah.
Claire.
Yeah, so the literature summary and aggregation part of ASGAD is really focusing on what's in the literature and what's already clinically associated. And so that's really post-bias, right? So we need to do better jobs of actually working with tools pre-bias and trying to find those nuances that are there. Within the context of that project, we've recently established a really nice collaboration with Google DeepMind, who have Alpha Genome, a really nice tool looking at variant effect prediction across both the coding and non-coding parts of the genome, and can predict regulatory variants. And so we're working with them to test and apply that tool in the setting of clinical genetics and trying to identify non-coding variants that, say, for example, cause a regulatory change that might cause disease. This is an absolute blind spot for us at the moment. Everybody knows these variants should be there, but we have no tools to meaningfully work with that type of data and make those discoveries. Some of these AI-based tools that are coming out recently are perhaps giving us the first threads to pull on to actually meaningfully do that in a genome-wide sense. So yeah, really exciting.
Well, we're moving into morning tea time, so I'm sure you're happy to talk with people throughout the day. Thank you very much. If you can join me thanking Kaz again.